We're going to go over some basics of DNA replication. The first thing to notice is that DNA in one human cell is 2 meters long. So in order to fit the DNA into the nucleus of a cell, we wrap it around proteins called histones in order to tightly package it into the nucleus of a cell. If we look at a chromosome before DNA replication, we see it as this single bar of tightly coiled DNA. After DNA replication, we see that we have the original bar, and at a centromere, we have a second bar attached to it, which is identical to the original bar. The second bar is the newly replicated DNA. At this point, we call this a chromosome, but we say that it has two sister chromatids, and these chromatids are identical to each other. Now, the term kinetochore refers to the proteins that hold the sister chromatids together at the centromere. Let's look at some basic ideas of cell division. If we take a cell with its DNA, and let's just look at one of these chromosomes. After DNA replication, we end up having a chromosome with two sister chromatids attached at the centromere by kinetochore proteins. So at this point, we have double the DNA, although the DNA is identical. Now we can do cell division, and we end up having two cells coming out of cell division with that original DNA that we had when we started. These cells can do their own DNA replication and do this cell division process again. Let's look at the apparatus that will cause the sister chromatids to be pulled apart. This is known as the centrosome, which is a structure of microtubules, and it has two centrioles that are at right angles to each other. These centrioles will extend what we call a spindle, which is a protein made of microtubules that extends from the centrosome and attaches to the kinetochore. Here is the image of it. We have a centrosome on the left, a centrosome on the right, and you can see that the spindles are reaching out. These spindles made of microtubules reach out and attach to the proteins of the kinetochore. After they attach to these proteins, they will pull the two sister chromatids apart. So now we want to go over the cell cycle and mitosis. We're going to actually start with a cell that needs to divide. The first part of cell division is the mitosis part. And this is where the DNA gets separated into two components. The second part of cell division is known as cytokinesis. And cytokinesis is really where the cells themselves split into two daughter cells. Now, at this point, we want to trace what will happen to one of these daughter cells. So we're going to look at this one right here. And this cell will go into a phase known as the gap 1 phase. During gap 1, the cell grows, it builds molecules, and it makes organelles. So we can see here that it is going through that gap 1 phase, which is abbreviated as G1. After G1, the cell has the option to go into either a G0 or gap 0 phase, or to proceed to the S phase or the synthesis phase. Let's first talk about what G0 or gap 0 will look like. Here the cell goes into a non-dividing phase, but it continues basic cell functions. So in the G0 phase, it does not have the objective to divide. If it does want to divide, it needs to proceed to the S phase or the synthesis phase. This is where the DNA repairs itself if there are any mistakes in it. And very importantly, it is where the DNA replication takes place. And so when we look at this cell after S phase, we see that the DNA is doubled. Since the cell wants to divide, it will also duplicate its centrosome. After synthesis phase, it will go into this gap 2 phase, or the G2 phase. So the G2 phase is where we have continued growth of the cell, and the cell assembles the proteins for cell division. And so we can see that after this G2 phase, that the cell is larger and is ready to divide. And here we can see that the cycle would start all over again, where we would go into mitosis, followed by cytokinesis, and then we could trace the cell cycle of another daughter cell.